Stop right there, Joker. You can't get away with this. On the contrary, my dear, all the sugar will be mine, and then I will control all the energy and food in Gotham. Joker, you fool. You can only use sugar to get a minor amount of energy. There are other molecules needed to survive. What? It's true. I need more protein to keep up muscle mass. And I need more lipids to keep warm. Sugars is the only thing that keeps me fully energized. I require a lot of nucleic acids to make lots of DNA. Too bad for you, you didn't get all the sugar. Now, Joker, the joke's on you. Oh, I miss Batman. Oh, Joker, I guess he couldn't get away this time either. Maybe next time he'll be hanging around for a while. Anyway, today we're going to be looking at the 4 macromolecules that are necessary for life. We've already seen one before in the form of glucose, which is a carbohydrate or sugar. We're going to look at the other 3 today and just have a general overview of what they are. Let's take a look. The 4 macromolecules for life. What are macromolecules? Macro means large. They are polymers. Macromolecules are large molecules that your body takes or organisms take from an outside source, breaks them down, and then puts them into new forms that are usable to make the organism survive. These 4 molecules are in large molecules that are found in all known forms of life. Now you'll have to excuse the lack of as many pictures in this one as I don't have internet, so I can't pull some off immediately for you. However, I'm able to show you some of the demos for it. Now the 4 macromolecules that we'll be talking about today, these are organic molecules that can be found in all of your foods. Now many of you may say things like calcium or salts are also important as well as water, but those aren't organic molecules. Those are just minor molecules that are used in your body. These ones are made naturally and used on a regular basis. The first one we're going to take a look at is lipids. Lipids for energy storage and cell membranes. Lipids, or more commonly known as fats, can be found all over the body as a rich form of energy storage, which we'll look at later. To feel what your lipid looks like, take your arm and grab a little bit of skin right here below, and then pull. This loose part is fat. We commonly see it in things such as oils, butter, olive oil, and other such materials. They're known for their rich energy storage. Carbohydrates for cellular energy. Carbohydrates are the next one. Carbs are infamous for giving us lots of energy. Commonly we see them in the form of sugar. Now this sugar is actually larger than what normal carbohydrates look like. In here, this fine granulated stuff is actually made of 2 sugar molecules, glucose, which gives your body energy, and fructose, which causes it to be sweet. There are many other types of carbohydrates out there that we'll see later, ranging anywhere from maltose to sugars and milk. But let's move on to our next molecule. Another such form of sugar that we may not be aware of is starch. Starch is this chalky powder stuff that you see that you typically use in baking and other such types of cooking. You often see it in breads, cakes, cupcakes, and other such. That's why those foods are considered to be high in carbs, because a single thing of starch is actually many, many glucoses long. Nucleic acids for cellular information and protein synthesis. Nucleic acids are basically what's in DNA and RNA. DNA is the molecule that makes you and your cells themselves. It allows it to store and transmit information, and we'll see it's a very complex process. But when you eat food, you're basically taking the DNA that's within those cells, for example steak, ripping it apart into tiny little pieces, and then incorporating it into your own cells. Proteins perform all necessary cell tasks. The last molecule that we're looking at is proteins. Proteins come in a variety of forms, in this case, miscellaneous deli meat. Proteins allow for your cells to do every little function, from building muscle mass, to breaking down molecules, and to taking glucose and turning it into rich energy. Proteins do everything, and we'll see that there's a special class of proteins later that are known as enzymes that allow the body to be able to perform these functions and allow you to be able to function at a high level. Your lab is going to be tonight, while at dinner, try seeing if you can identify what you're getting out of each of your food. For example, looking at a nice tasty steak or piece of chicken, you know outright that you're getting the protein. However, is there anything else within that for the 4 macromolecules that you might be getting out of? Look forward to talking to you tomorrow. Macromolecule Identification Lab, a handy technique for identification of different macromolecules present in foods and cells. Bring in one item of food. The food can be larger, but we will cut it down. Step 2. Make sure that you have a test tube holder at your station. Step 3. Grab 6 test tubes from the front of the class in the bin marked clean and 2 pieces of paper. 4. For controls, take one of each of the following prepared dishes: carbohydrates and sugars, lipids and fats, starch and large sugars, and proteins. Add each of the above samples except lipids into their own test tubes. You should have only used 3 of the 6 test tubes at this time. In the sugar test tube, add 3 drops of Benedict's solution. It should change to an orange or reddish if glucose is present. In the starch test tube, add 3 drops of iodine. A black end color will indicate starch is present. In the protein tube, add 3 drops of burette solution. It will turn purple in the presence of proteins. 9. Rub lipids onto the paper bag, then hold the paper bag up to the light. If lipids are present, the bag will become translucent, meaning you can look at light through it. Now divide your brought-in sample between the 3 test tubes, saving a bit for the paper bag test, so 4 total. Repeat trials and record which molecules were present in the food that you brought in. Okay, so here's how you're going to be working your lab for today. First off, some of the equipment that you're going to see. Here is the test tubes that you're going to be using. Notice I have 6. This holder is just a tape deck. Many of you will have a test tube holder, but 2 of you will have these test tubes. The tape deck. The solutions that you're going to be using are these 3. This blue one, which is Benedict's solution. This off-color one, which is burette's solution. This black one, which is iodine. They'll all be properly labeled. You'll also have 1 paper bag, as well as a thing of starch, a thing of oil, a thing of sugar, and 1 small sample of protein. First one we're going to test is the sugar. Take your scoop and scoop out a little bit of the sugar. Then add it to a test tube, so it rests at the bottom. Taking your blue Benedict's solution, suck up a little bit and add to the test tube. In the presence, we should see it turn a darker blue. You should end up with a result that looks something close to this. That indicating the presence of sugar. The next one we'll try is starch. To make sure that there's no contaminant, make sure that you've slightly rinsed your scopula. Then scoop up a small bit of the starch and add to a test tube. Tap gently to make sure that the starch goes all the way down to the bottom. Then take your iodine solution and add it. Remember, only 3 drops would do. 1, 2, 3. Shake it up, and then hold it up to the light. You'll notice that the iodine has gone from brown to black. This indicates that starch is present. Let's do protein. Taking your scopula, scoop up a small sample of the protein that is supplied. It may be a little bit tricky. You may have to use your finger. That's okay. Like me. Ta-da. Then add to your test tube. Once again, give a gentle tap on the bottom to make sure a little piece of it goes down. Then take your burette solution and add. Notice the color. That chalky color, or it would indicate it. You also should maybe see a red or an orangish color present. Lastly will be your oil test. Take the hilt of your scapula and dip it into the oil that's provided. Then, rub it gently onto the paper bag supplied with you. You may spread it around with your finger if you want to get the texture down. And then hold it up to the light. If you can see through the paper bag some of the light that's coming through, or it being translucent, then that means that there's a lipid present. In this case, yes. You're going to repeat these 4 experiments, only this time using your food to determine what's inside of it.